Hello everyone, today we're going to talk about the identifications of autophagy-related genes and the prognosis of oral squamous cell carcinoma. Oral squamous cell carcinoma is the most common histological subtype of oral cancer. But over the past year, there has been no improvement in the overall 5-year survival rate, and the recurrence worsens the chance of survival. Furthermore, tumor node metastatic classifications fail to assess the clinical outcome. For these reasons, it's important to find a new predictive biomarker for patients. In previous study, there has been shown that gene expression is associated with the cancer prognosis, while autophagy is associated with the development of oral squamous cell carcinoma. In this study, we're conducting a systematic analysis to explore the relationship between the expression of autophagy-related genes and the prognosis of oral squamous cell carcinoma.
A list of 232 human autophagy-related genes was obtained from the Human Autophagy Database. The TCGA database provided the transcriptome data, while patient clinical pathological data was obtained from the cBioPortal database. The study involved 331 OSCC patients with survival information. Prognosis-related genes were validated using gene expression profiles from the Gene Expression Omnibus database. The transcriptome data underwent log2X+1 transformation. The Wilcoxon-Sai rank test was applied to identify differentially expressed genes. The threshold for significance was determined based on Bonferini correction and full change.
We conduct GO and KEGG enrichment analysis to annotate the genes. GO analysis explore biological function, cellular component, and molecular function, while KEGG provide additional insight. We identify 33 genes as potential prognostic marker using unique variant congregation, and we use Kaplan-Meier plot and congregation to analyze overall survival.
OSCC cell lines along with the normal cell lines were obtained from the cell bank of the Chinese Academy of Sciences. These cell lines were cultured in DMEM, supplemented with fetal bovine serum, penicillin and streptomycin, and then incubated. The tissue microarray of OSCC was prepared by Shanghai Aotu Biotech Company. The microarray consisted of 18 OSCC tissues and 5 normal tissues, which were incubated with primary and secondary antibodies. Their staining was semi-quantitative and based on the extent and intensity. The magnifications used for evaluation were 100x and 400x.
For reverse transcription and quantitative PCR, we utilized RNA-seq minikit to extract total RNA. Then it was reverse transcribed into cDNA using cDNA reverse transcription kit, and we employ the R software for all statistical analysis, and we use Kaplan-Meier plot and congregation to analyze overall survival. For the result part, we analyzed RNA sequencing data using Wilcoxon-Sai rank test, and we successfully identified 33 genes consisting of 24 upregulated and 9 downregulated genes. We conduct GO and KEGG enrichment analysis to explore the biological function of the genes. The results show that the genes were related to autophagy, focal adhesion, and receptor ligand activity. Furthermore, they were found to be enriched in apoptosis and viral infection like HPV.
From the complete question analysis, the high-risk gene will be ID and BRCA1, and the low-risk gene were NKX2-3 and SPHK1. The dot plots show that the patient with high-risk score had higher mortality rates and shorter survival times than those with low-risk scores. The results of GSE 85195 reveal the differential expression of BRCA1, NKX2-3, and SPHK1 between tumor and OL tissues. Similarly, in the analysis of GSE 10121, BID, NKX2-3, and SPHK1 exhibited differential expression between tumor and normal tissues. In OSCC cells and tissues, SPHK1 showed upregulation while NKX2-3 demonstrated downregulation compared to normal controls.
Congregation analysis identified 4 genes linked to OSCC development: BRCA1, BID, NKX2-3, and SPHK1. SPHK1 is overexpressed in OSCC tissue and associated with patient survival. NKX2-3 is implicated in cellular differentiation and has a high risk associated with alloleukopenia. BID and BRCA1 correlate with poor prognosis in thyroid cancer and OSCC respectively. However, further research is needed to understand the exact mechanism of SPHK1 and NKX2-3. in OSCC. Study limitations include a small sample size for normal tissue analysis and a lack of treatment information from most patients.
In conclusion, 4 genes that are associated with the prognosis were found. Notably among these genes, SPHK1 and NKX2-3 were differentially expressed between oral squamous cell carcinoma comparing to normal. These findings highlight the potential of these genes as promising biomarkers for oral squamous cell carcinoma prognosis.